ఏంద్రా అమ్మని ఆపరా బాబు. హలో సుష్మా.
Yes, sir.
అమ్మా స్టార్ట్ చేద్దామా?
Yes, sir. Can I start, sir?
Yes, ma.
Good afternoon. Welcome to the day 14 session 2 of the workshop. Our session invited speaker is Professor Sriram Krupanidhi, Head of the Department of Biotechnology, VIGNAN'S Foundation for Science, Technology and Research, Vadlamudi. Today he is going to give his guest lecture on vaccine research. Sir, could you please? Sir, your mic was muted.
Yes, ma. Sushma, you can read his profile also before introducing to the participants can know his profile. Okay. Before Rupanidhi sir starts, you can read his profile. Please wait, Rupanidhi sir. Are you ready with the guest profile, ma?
Yes, sir.
Okay, okay. Slowly you can, no problem. You can start slowly.
Okay, sir. The interest in teaching and research made Professor S. Krupanidhi to get attached to the profession since the year 1975 as research scholar and followed by the faculty in Sri Satya Sai Institute of Research Technology. After superannuation, he took a professor position in VIGNAN'S Foundation and Science, Technology and Research University. Since the time he was inducted in the chosen field, he made persistent efforts to unfold the physiology of dormancy among gastropods and unravel the quantitative parameters related to the oxygen affinities of hemocyanin and its immunological attributes. He was widened the areas of, especially in the areas of interest, after assuming the teaching position by attending the workshops at the University of Pune, Shivaji University of Gwalior, and BARC, Mumbai, IIC Bangalore, Department of Biophysics, University of Madras. He organized several national and international conferences and also training sessions to train teachers in the field of developmental biology, immunology, and molecular physiogenetics. His contributions in biotechnology is in the field of green environment, wherein his group developed a prototype for the biotransformation for domestic vegetable scrap in useful compost rich in mineral nutrients, and the same birds field for patent. With a wide experience in teaching, he contributed popular science articles and a monograph on nutrition and health awareness, and also wrote the online book on fermentation technology instant class notes. He presented his contribution in the field at various international conferences in Karolinska Institute of Medical Science, Stockholm, Biopolis, Singapore, and Hong Kong Baptist projects on the topic molecular affinities of hemocyanin of gastropods. Professor S. Krupanidhi is currently deeply involved in rebuilding the classification of Indian gastropods, employing the tools such as the multiple sequence of similarities of COI 16SR DNA genes. He is fully satisfied with his profession as a teacher, and also his laboratory learning tools were developed for the student to adopt the learn, such as the demonstration of polar body cleavage division and habituation learning, construction of oxygen equilibrium curves, PRL program of biological students with 14 exercises, molecular phylogeny, using tools such as MEGA, POP, RASMOL, develop a technique using hemocyanin and antigen to develop crescentic arcs employing outcrony double immunodiffusion technique. Developed a protocol for the bioconversion of vegetable scrap into the compost. Causal loop diagrams in designing the metabolic systems and also enzymatic response to develop antibodies in vitro.
Yes, yes, excellent, excellent. To his credit, he has completed so many research projects and funded by government agencies like UGC, DST, DST FIST. He acted as a DBT PF coordinator, SAP DRS level 2 coordinator. He has so many sponsorships. Currently, he has one sanctioned projects such as phylogenetic analysis and barcoding of Indian apple snails as a prelude to their conservation strategy. He has published more than 200 research articles, including book chapters in high impact factor journals, and is also an Dr. G. Subbaraju Memorial Awardee. It was presented by AP Academy of Sciences for the best article published in the proceedings of Andhra Pradesh Academy of Sciences, Hyderabad. With this note, I would like to welcome Professor S. Krupanidhi to present his issue.
Thank you, Abraham. Thank you, Hima Sushma, for the generous and elaborate introduction about me. Now, for the time being, what I wish to let you know is I'll put off my video so that there may not be any interruption in my teaching, okay, in my lecture.
Sure, sir.
So, good afternoon to all participants, experts, and students, faculty who are actively involved in this workshop for the past 14 days. Along with you, in most of the sessions, I'm also participating and learning in this machine learning tools. And now I'll be handling for you all web-based tools for vaccine design. So this is also a little bit related to machine learning. And here, our interest is how a vaccine can be designed using some of the immunoinformatic tools. So vaccine, little bit let us know what the vaccine is. Vaccine that has come into work since the time of Edward Jenner and Louis Pasteur, and it is for prophylactic. Nowadays, therapeutic vaccines to cure cancers also have come in 2018. Tatsuko Honjo and also the other scientists from Texas A&M University has come out with a few immunoglobulins like ipilimumab to cure colon cancer and other cancers. Okay, ultimately vaccine is for prophylactic. And hitherto, for the vaccine to design, it used to take good amount of time. Now all that has been altered, changed, modified, made very fast, that we are now experiencing in our COVID-19 vaccines like Covaxin, Covishield, and several other Sputnik V and all those things. Now let us see a few of these things in related to immunoinformatic tools. So the main objective of my talk are two. One is acquaint with the complex immune system. So without knowing the immune system, we cannot get into the vaccine. So let us know the complex immune system in brief, because immunology itself is a wide subject. Here I'll be handling in 4 to 5 slides. And the need for web-based tools to design vaccine. Abraham, if my talk and audio is not well, please remind me in between. So we are going to a specific example to let you know how we are going to explain the bioinformatic tools. Hitherto we went through many, what is it called, demos. So we have gone through this TensorFlow and several other things. And here also I will show not a demo, but a few slides which have been worked through those immunoinformatic tools. Okay. So ultimate outcome of this presentation will be, you will be knowing by the end of this talk, the acquaintance with the web-based tools for epitope-based polytope design that will be known to you. The case study, an experimental project study that we have done design of polytope using receptor binding domain, orbital region of spike protein of coronavirus. And these things will be known for young participants to plan their projects in a similar way. So now the contents of my presentation will be as follows. So to begin with, vaccines and their need. A few slides later, brief view of immune system. Next is MSC1, major histocompatibility complex 1. So there's MSC2 also. We are not going to consider here MSC2 because the virus proteins are being handled by MSC1. Okay, and online software tools, 1 or 2 slides, a need for computer-aided vaccine design, computer-aided vaccine design, and ultimately one example that we're working, the spike protein of coronavirus. Okay, the vaccines and their need. So still there are good many number of diseases for which vaccines have not come. Okay, so as bioinformatician experts, and it is our as biologists, it is our interest to get to know what are the complications, what are the components involved in designing the vaccine. Once you know, there are plenty of companies are coming up in India about the vaccine development, particularly for COVID-19. Hitherto in the COVID-19, we got one inactivated vaccine, that's what called Covaxin. The other one that has come from the Serum Institute of India is called Covishield. That's vectored vaccine. It's a new type of vaccine, vectored vaccine. Okay, so far recombinant vaccine has not come. Okay, in addition to that, there is a talk around that there is a viral escape. That is, the virus is changing its, what is that called, amino acid sequence. As a result, now in the news, there is, people are telling N440K, something like that. So vaccines are emerging field, and it is a continuous field, it is an ongoing field, wherever it is required for the human welfare, Let us see now. Now, if you go through this particular map, see, a few I'm showing, not all, what is it called, viruses, and majority of the viruses have emerged from these African countries and also in Asian countries. So if you see a few examples, let us see Zika, yellow fever, Rift Valley fever, and chikungunya, and HIV, Ebola, and MERS in the Middle East, and dengue, and SARS, and Japanese encephalitis virus, and novel coronavirus 2. So they all have taken their origin, they're erupted in this region, and that is spread across the world. It's a different issue. So like this, many of these viruses are having their origin here. Apart from that, because of our lifestyle, that is, we all move as a flock. So as a result, the virus is neatly spreading from one individual to the other individual, and it is undergoing a lot of mutations, and generations of generation, it is surviving in collaboration with human host. Okay. Now, if you take up the vaccines, the vaccines so far have been made only for 36 diseases, viral diseases. Okay, so there are more than 400 human pathogens. For all those things, there is a need to make the vaccines. So there are inactivated vaccines, live vaccines, and toxoid vaccines. Okay, now if you go through a few, we are going to come across recombinant vaccines. So that's what the recombinant vaccines for COVID-19 yet to come. It has not yet come. Okay, so where the recombinant vaccine is made, it's much of genetic engineering techniques and also bioinformatic techniques. Okay, so that's what these many vaccines are through. And also in this list, there is no, what is this called, our coronavirus. Coronavirus vaccine is also through now. Now, this is the deaths because of lack of vaccines since the year 2002. So now this situation must have been a little bit developed. But with regard to the coronavirus, we are facing a lot of pandemic, world over. So now let us come to the point now. That is a little bit introduction point. Okay. So now if you take up a little bit, every one of you know about the coronavirus now. Coronavirus is having around 30,000 base, approximately 30,000 base pair nucleotides. Okay. And is also having around 15 to 16 proteins. Okay, and those proteins, if we take up into consideration the what is potential epitopes, so there will be 175,000. Like this. There's so many epitopes will come. So many epitopes. I will let you know over the time. Okay, so those epitopes have to be again processed through bioinformatics tools to have consensus epitopes. Consensus epitopes. Okay, so they will be around 2,000, approximately 2,000. So from there, if you have to come down to confirmed epitopes, okay, from this confirmed epitopes, we have to come down to virulence epitopes, those epitopes which are virulent. Okay, so from this potential epitopes, which are getting anchored, that the computer tools, IEDB, that is epitope database, immunoepitope database, IEDB, show this many potential MSC1 binding anchored epitopes. By the time we process those things, it will be 3 to 4 virulent epitopes will come. So like this, all this involves good amount of bioinformatic tools, wherein we have to get to know what is MSC1. So MSC means major histocompatibility complex. So the histocompatibility complex from individual to individual, it varies. As many stars in the sky, so many types of MSC class 1 molecules that are present with us. So there is every need for us to evaluate these epitopes in combination with MSC molecules 1. Okay, now. So now there is reverse vaccinology. The reverse vaccinology has now come up into the field because for a regular conventional vaccinology, we have to culture the cells, microbes, virus. Okay, and ultimately after cultivation, we have to make them killed or inactivated. And that later we have to go for clinical trials. All that takes nearly for 15 years. Right, so now in reverse vaccinology, genome-based approaches, and again, the genome variations within the species is called pan. It will take nearly 1 to 2 years. And structural vaccinology, where these recombinant vaccines are being given, just a spike protein or hepatitis B surface antigen. So hepatitis B surface antigen is a recombinant vaccine. Okay, so like this, determination of the antigen structure, particularly if we take up COVID-19, we have got the receptor binding domain. So that particular aspect can be given as a vaccine, wherein the vaccine, because the preparation of the vaccine also will get lowered in its cost. Okay, so like this now, the focus is much on the reverse vaccinology, for which bioinformatic tools are very much essential. Okay, now as you see in the previous slide, there are live attenuated vaccines, inactivated vaccines, toxoid vaccines, and subunit vaccine is the one that we are planning through bioinformatics. Here, bioinformatics scope come. In inactivated, in live attenuated, in toxoid, bioinformatics scope is meager. Okay, now recently the vectored vaccines have come, and wherein the genetic engineering tools and bioinformatic tools are involved, Covishield and Sputnik. And with regard to the mRNA vaccine is again genetic engineered based tools, and again the bioinformatic based tools. Okay. Now the subunit vaccine means if we had to work with what is called the gene sequence. From there we are going to have the protein sequence. Okay. And from this protein sequence, if we get into the IEDB, immunoepitope database, we'll be getting, though the sequence is this much, we'll be getting thousands of epitopes, of which we have to select a few peptides. Epitope means anything but they are peptides. and those are the peptides which are having virulence factors. Okay, so they had to be selected. So all that will be done through our bioinformatic tools. I will be showing an example at the end. Okay, now, so whenever we are having, there are good many number of strains have come in COVID-19 also, coronavirus. So like this, let us see an example here. The equivalent of this epitope-based vaccines is to select epitopes in a way so that they together cover all strains. Okay, so now let us take up, let us say this is an epitope having 9 or 10 amino acids. This is strain 1 and strain 2 of a particular virus. The strain 1 is having 4 epitopes here. Strain 2 correspondingly does not have here. It may have somewhere in the other regions. Okay, if you make a vaccine, if you make a polytope for these 4 epitopes, it doesn't take into consideration the strain 2. The strain 2 we cannot protect. We cannot get protected from strain 2. Okay, so like this, if you have even coverage of strain 1 and strain 2 epitopes. This is the strain 1 epitope. This is the strain 2 epitope. Both epitopes composition, if you take up and make a polytope and prepare a vaccine formulation, then that vaccine that is made takes care of the infections against strain 1 and strain 2 of coronavirus and anything. So that's what the polytope vaccines which we prepare contain the epitopes of the different strains, so that we get protected, so that there will not be the spreading of the communicable diseases in the society. As a result, the health of the community, the health of the family, and ultimately the wealth of the nation will be taken care of. So that's what we all have to promote our kith and kin to have the vaccination. Clear? Now, so whenever we take a vaccine like this on the 0 day, then there is a lag period. See, for about 5 days or so, nearly for about 5 to 6 days, there is no antibodies developed. There is no antibody, there is no memory cells, B cells, T cells will not be developed. Still at the region of the dendritic cells, and dendritic cells coupling with MSC molecules for antigen presentation, all that keeps going on here. Okay, then once the T cell encounters that dendritic cell, then the T cell influences the B cell. B cell produces immunoglobulins, antibodies in the serum. They get enriched in the serum. Okay, so it is only one-time memory, one-time primary injection. So then this is being followed by about soon after the first month of the primary immunization. The secondary immunization is given. The secondary immunization, there is no lag period. So we are seeing here the lag period. The lag period is missing here. The second immunization. So soon after getting the second vaccination, our values, that is immunoglobulin values, keep increasing in the serum, so that we are highly protected. So that's what going on here. That was shown in this diagram. By chance, again, if we take along with the second, there's one more vaccine. That's what kids are given, different types of vaccines against what is it called, typhoid, against salmonella, and all those things. And the second vaccine is given. Second vaccine also take a lag period and the response period, immune response period. That's what here there's a lag period and there is an immune response. And again, the arousal of immune response for the second vaccination is at its peak. Okay. Now, so there is always viral escape. So that's what we are continuously facing now. So as a result of the viral escape, and our these vaccines also have to take into consideration, for which I have shown you earlier, evenly taken epitopes from strain 1, strain 2 will give us protection from the different type of the viral escapes that we are encountering in the society, in our community. Okay, now, so this is a little bit about the need for vaccination, the need for vaccines to develop. Okay, now let us see the brief view of immune system. Okay, so the immune system, as you all know, it is, if you take up the whole body, if we are weighing for about 80 kgs, immune system does not weigh even for half a kg. Not even for nearly 250 grams, it will be 250 grams. Okay, so there is other systems in the body which are weighing kgs. Okay, so they are not protecting your health. Your muscle is not protecting your health. Your brain is not protecting your health. Okay, your heart is not protecting your health. And it's only the immune system is the defense system that is protecting the whole of your system, the whole of your body. Okay, so as a result here, innate immunity. The innate immunity is the one which we got inherited over the generations. It's not only from the human beings. In the evolution, a few of the features we inherited. Okay, let me say a few examples. Particularly, there is TLR, toll-like receptors. Okay, we are having the toll-like receptors on all our leukocytes. Those toll-like receptors identify the pathogens. Okay, so that is given to us evolutionarily. Okay, that is one. The second thing is, there's amoeba. Amoeba is having pseudopodia. That is the ancient specimens amoeba. So even we are having a few of our leukocytes, they're amoeboid, dendritic cell. Okay, so these are the dendritic cells which are the most powerful, what is that called, and pathogen recognizers. They recognize the pathogen. Okay, and they process the pathogen. Okay, the antigen presentation is being done by this dendritic cell. Okay, that's what is all involved in the innate immunity. In addition to, there are several other features like skin, mucus, or other things are there. But at molecular level, I'm telling, toll-like receptors and this amoeboid type of cells that are there with us, they are processing innate immunity. They are the part of the primary sentinels in our system, innate immunity. So in this, innate immunity is pre-existing. That's what I was mentioning to you. It is inherited to us through the evolution, and it is non-specific. That is, it doesn't respond specifically for one type of the disease or one type of the pathogen. The response of the innate immunity is uniform for all type of the pathogens. It doesn't specifically give more emphasis for corona, gives least emphasis for what is it called dengue, and gives still more least emphasis on malaria. No, it's what is it called? Its activity towards this any of the pathogen is uniform. That's what it's non-specific. Okay, adaptive immunity. Adaptive immunity is the crux for our vaccine development. So scientists, those were the days, knowingly or unknowingly, there is an adaptive immunity. They started this curing of smallpox from cowpox. Okay, so like that, the people have understood what is this adaptive immunity. So this is the basis for our vaccine, adaptive immunity. Let's say in this, wherever active immunization is given, so that whatever the product that you are giving in the vaccine, let's say the protein or an inactivated virus, then we are making our system to learn what is this protein, what is the epitopes that are present in the protein. So ultimately, this vaccine is making us to adapt. The vaccine is making us to adapt to a pathogen. So that whenever we will be learning about the pathogen, our system learns. Okay, and it's not the brain learns, our immune system learns. So that's what this adaptive immunity. Now in this, it is response. Okay, and the adaptive immunity, it responses to the pathogens. That's what now, if I get infected pathogen, if I am vaccinated, if I get infected with the pathogen, immediately our system responds to arrest the growth, budding and proliferation of coronavirus. That is response. Again, this adaptive immunity is highly specific. It is highly specific. That's what now people are worrying about the vaccines. And a few of the vaccines are inactivated coronavirus. But a few of the vaccines are only the spike protein are given in the vectored vaccine. So if the spike protein totally alters, then our vaccine doesn't work. We have to take again other conserved proteins of the coronavirus as the vaccine. That's what the adaptive immunity, the vaccines which are developed, they are highly specific towards the pathogen molecular structure. So now let us describe this diagram to understand more about the innate immunity and adaptive immunity. So in the innate immunity, what is happening is the microbes, they have got their own routes to get into our system. like nasal routes or oral routes or any wounded skin, they will get in. Once they get in, they go into our system where there are phagocytes. These phagocytes are called these dendritic cells, macrophages, monocytes, and these dendritic cells they engulf and communicate to the complement system. There is another thing, communicate. Okay, and later there are a few of these microbes. They are floating. They'll be handled by the NK cells, natural killer cells. They will handle it. Okay, so once these phagocytes process this antigen that is being presented to the T cells, T lymphocytes. Okay, the T lymphocyte carries it and it gives the information to the T effector cells. T effector cells later presents to the B cells, B lymphocytes. Okay, the B lymphocytes later develop the plasma cells, and the plasma cells and memory cells. The plasma cells give antibodies. So these antibodies neutralize. These antibodies neutralize this pathogens. Proteins of the pathogens or pathogen itself or structural proteins which are present in the surface of the pathogens get neutralized. Once they get neutralized, then the pathogen is made immobilized. It cannot further, what is that called, penetrate into the other host cells, So like that, the adaptive immunity and innate immunity both work together. Though the adaptive immunity comes into action after innate immunity. So this is the adaptive immunity principle that is being adopted in the development of the vaccine. Now, let us see a few of this immune system. We've got the phagocytic cells. As I've been mentioning, dendritic cells. They're the best antigen presenters. Of course, macrophages, monocytes are there. They're all related. And we've got the lymphocytes, B lymphocyte, which is having the memory. And all these memory cells are present in the lymph nodes, not in the bone marrow. Lymph nodes. Bone marrow is the, what is it called, hemopoietic system. Hematopoietic system, where from the naive B cells are released. And once the B cells are released, they get into the lymph nodes. The lymph nodes is the place where the blood flows through and they get exposed to this type of the pathogenic epitopes. T lymphocytes, there is T helper cells, T cytotoxic cells. Again, the T helper cells, Th1, Th2. There will be pre-inflammatory and anti-inflammatory, post-inflammatory, several things are there here. Okay, now NK cells. NK cells are natural killer cells. They are not a sort of adaptive immunity. So natural killer cells, if they come across any cell that does not have MSC class 1, natural killer cell kills them. So that's what, that is which of the cell that doesn't possess MSC class 1. Those are the cancerous cells. Okay, cancerous cells priority is for budding only. They do not develop this architecture of MSC class 1, MSC class 2, all those things. So the expenditure for production of the MSC class 1 is waste for them. They wanted to divide. So in such a type, if those cancer cells are floating, then this NK cells, because they've got missed NK, there is MSC class 1 or depleted MSC class 1, this NK cells will be killing those cancer cells. That's what mostly it is anti-cancerous NK cells. Even otherwise, any bacteria comes, bacteria does not have our MSC molecules. Bacteria never contains MSC molecules. So it finds bacteria as foreign, so that NK cells will get activated and kill this bacteria. So that's what NK cell comes under this innate immunity along with dendritic cells. But these two lymphocytes, B and T lymphocytes, they come under this adaptive immunity. So let us see the cells of the immune system. This is what going on in the hematopoietic system, wherein hemocytoblast. And from this hemocytoblast, there are two lineages. One is the lymphoid lineage, the other one is the myeloid lineage. Okay, so the lymphoid lineage later gives the T cells, later natural killer cells, B cells. Okay, the T cells further gets all the start of the divisions and takes place only through cytokines. The cytokines are involved. So that it is going to become T helper cell, T cytotoxic cell, T memory cell. Like that, B cell is going to give rise the memory cell and the plasma cell. So this is what nowadays most of us are hearing in the news: plasma therapy, plasma therapy, plasma therapy. So plasma therapy contains the immunoglobulins and this type of the plasma cells, which will be useful for curing a new patient. Okay, so like this, these are all myeloid lineage. The myeloid lineage contains ELAs, toll-like receptors. Again, they are all amoeboid type. Amoeboid type. That's what these cells and these cells are shown in different way. Amoeboid type. They are involved in innate immunity. Okay, so particularly see the structure of the dendritic cell, which is more oriented towards endocytosing a pathogen. Okay, so that's what. So now again, the two divisions. Humoral immunity contains antibodies. Cell-mediated immunity contains the T cells. That's what. So now we'll let us, because without having the background in the subject, we cannot get into the bioinformatics tools. So now we are coming a little bit focused towards the bioinformatics. This I have been telling, this is the dendritic cell, a very good antigen presenting cell. Okay, it has become good antigen presenting cell because it feeds, it endocytoses pathogen. Once the pathogen is taken in, there is a proteasome, a mill which digests the protein through hydrolytic process. As a result, fragment of this antigen will be coming. Okay, so these fragments are presented through MSC molecules. Let us say for the time being, let us assume MSC molecule. Okay, now the question, please understand here, this is most important. Those of you who are interested to work with those are you who are interested to work with the immunoinformatic tools. Now let us see, from this pathogen, we are now shown here nearly 10 antigen fragments. In this diagram, we cannot show more, only 10 are shown. There will be thousands of protein fragments will come. So these are all the fragments. Okay, so that is cleavage sites of the proteins. So out of these fragments, a few can anchor, a few can bind to the MSC molecule. That's what called protein MSC complex, protein MSC molecule. The one that binds to MSC molecule, that is called, what is it called? Epitope of this pathogen, epitope of this antigen. So here again, there is one or two slides I have to explain and repeat for reinforcement. So these what is this called fragments are not getting bound. One or two are getting bound. They constitute the epitope. So that's what the epitope we have to what is it called? Find out through bioinformatic tools. That's what in the first diagram we have shown the potential epitopes which can bind to MSC1 molecule, a funnel-shaped diagram I have shown you. Later, I have shown you what is this called the confined epitopes, confirmed epitopes, consensus epitopes. Later, I have shown you virulent epitopes. So that's what here is happening in the, as it is in the live system. This is supposed to be the virulent epitope. This virulent epitope we have to predict through. This bioinformatic tools. Okay. Now, the next point I will let you know, another important bioinformatic tools which you have to understand here is MSC molecule. Okay. So you're all familiar what are the ABO blood groups. ABO blood groups, there are two types of alleles, and O1 doesn't have the allele. As a result, it's called multiple alleles. Now, with regard to this MSC class 1, A is diversified, it's having more than 500 alleles. B is having some other alleles, and C is having several other alleles. So it is the best example for the multiple allele inheritance, and again it's co-dominant. So like this, these MSC molecules are not the same in the two individuals. So this is what our even kidney transplantation time also HLA typing is being done. The HLA typing is done that the minimal HLA, what is that called, the variation they consider. They will not have total HLA similarity. Minimum HLA differences they consider for this. So for that minimal differences, they give all immunosuppressive drugs. Okay, that is a different issue. So here much of our bioinformatics work will focus on MSC molecules anchoring to this type of virulent epitopes. Okay, that's what. So let us see now, once the T helper cells is getting prompted, here again, see one more point here. This is called T cell receptor complex. So the T cell receptor complex accepts the linear epitopes. Okay, and then from this T cell, there is the prompting to the B cell. B cell at times accepts what is the conformational epitope, discontinuous epitopes. So that's what here, these things we have to discuss at length in the coming slides. Okay, so ultimately this is what the humoral immunity wherein antibodies are involved, and cell-mediated immunity where the T cell is involved. So in the immune system, those of you who are not that much aware of the immune system get to know that the T cell is the master immune cell. Okay, the dendritic cell is said to be the master antigen presenting cell. Okay, the B cell is said to be the memory cell, which gives good amount of what they call immunoglobulins. If there is, see, if you are having dendritic cell, if you are having the T cell, if there is any mutation with regard to the B cell, that's what we see ADA, adenosine deaminase, a sort of a mutation in the B cell. Okay, then the B cell does not produce immunoglobulins. If there is no immunoglobulins, we are having immune deficiency, natural immune deficiency. That's what there is a situation called severe combined immunodeficiency SID. Okay, so as a result, that can be genetically repaired so that the B cell is made, because it's all happening in hemocytoblast cells. The stem cells can be repaired and again reinfused. So that's what's been done in one case in Sri Lanka. Okay. So now this is again the pathway to show to you. This I'm interested to show to students. Whenever the antigen comes, the dendritic cells process them, and sometimes if antigen is small antigen, B cell directly takes. So if antigen is so big, then T cell is involved, and here IL-1 is involved, here IL-6 is involved. As a result, B cell prompts. How long the B cell produces immunoglobulins? There is a feedback mechanism. And these immunoglobulins, if there's no question of encountering an antigen, there's no question of encountering a pathogen, then they have to undergo catabolism. Okay, and this catabolism leads the B cell not to produce any more immunoglobulins. And again, there's a T suppressor cell feedback mechanism. This T suppressor cell promotes not to activate the B cell, not to activate the T helper cell, not to activate this macrophages. So like this, there are several pathways that the participants is better to understand. Now, the immunological memory. The immunological memory is more rapid. Okay, so that's what if you take vaccine, will get protected from any of the pathogens. And the immunological memory is more intense. Okay, the immunological memory is more prolonged. The kids of 2 years, 5 years, 1 year, they are taken the vaccine, throughout the life they will not get what is called either chickenpox, mumps, and all those things. They will not be facing. And this immunological memory is highly specific. So hitherto, our population has never experienced COVID-19 or novel coronavirus. That's what we do not have the specific immunity against the coronavirus. Now that's what if you take the vaccine, we develop the specific immunity. Not only for those were the days we remember that vaccine is given only for small kids of less than 5 years. Now above 60, above 70 years people also vaccine is given. That means they have got the hemocytoblast cells, hematophytic environment wherein the stem cells are there. They will develop what is it called the plasma cells and ultimately the memory. That's what more specific. So now this is another good concept for you all to follow. This is Nobel laureate explanation of McFarlane Burnett, and she explained this antigen that comes through this epitope. We have got several of the B cells. We do not have one B cell. We've got several B cells, of which of the B cells that encounters this antigen, epitope presentation by T helper cell. T helper cell presents this antigen. So that B cell undergoes proliferation. And ultimately it gives the it bails out the immunoglobulins. Okay. So like that, these immunoglobulins are just now being released to fight with the pathogen. That's what clonal selection theory proposed by McFarlane Burnett. Okay. So now, so we have gone through the brief immune system. Ultimately, in all these things, we focus how the immunoglobulins are being released, what is that antigen presentation, what is that MSC molecule role, how does epitope binds to this. All fragments of the antigen do not constitute the epitope. Whichever that anchors to MSC molecule, that constitutes epitope, viral epitope. That MSC and epitope fragment, that complex is being recognized by T cell receptor complex. If there's only MSC alone, T cell doesn't approach the dendritic cell. If there is MSC along with anchored, what is that called, this epitope, then the T cell recognizes it, the T cell further processes it and communicates to the B cell. So all that we have gone through in the previous, a brief overview of this immune system. Now let us see the MSC class 1. Why? Because we are mentioning about the MSC here is, without MSC knowing, having the knowledge of the MSC only makes us understand the bioinformatics. Just like that, we cannot operate the bioinformatics tool, because what is the role of MSC? What is the population coverage? What is the population of this Andhra or Tamil Nadu is having the MSC haplotype? That has to be studied to begin with. We should have the understanding. Then only we can develop epitope. Then only we can develop the polytope. Then only we have what is called vaccine design. So that's what MSC class 1 is taken into consideration here. There is MSC class 2 also. MSC class 2 is basically for bacterial proteins. Now we are focusing on viral proteins. We are speaking on the MSC class 1. Okay. Now, so this is a cartoon to explain to you again in a similar way what we have done earlier. So let us say this is a foreign protein. It's a foreign protein, viral protein. So viral protein, let us say our spike protein is having around 1200 amino acids. So let us say this is 1200 amino acids protein. If this comes into the cell in the proteasome, it gets fragmented into peptides. How many types of peptides? This 1200 amino acids will be cleaved into different stages. Okay, by different proteolytic enzymes. As a result, we'll be getting varieties of the fragments. Okay, a few of the fragments go and they get reaggregated because the peptide does contain only amino acids. Amino acids are the building blocks of proteins. They get aggregated. Even viral proteins are being used for building our protein structures. Like that, they get reaggregated. A few of them will get recycled, and those are the peptides which are virulent will be taken by transport-aided proteins, transport processed proteins. Okay, and this tap is present over the endoplasmic reticulum. So once it goes, wherein the MSC, naive MSC will be there. Along with the naive MSC, they get attached. Okay, this is the peptide. It gets attached here. So this is taken out. MSC molecule is taking out the epitope. Once the epitope is presented by this dendritic cell, killer cell, T cytotoxic cell comes and identifies and does its action. So this is the function of the, what is it called, inside the cell, what is happening. So here we have to understand the epitope properties, physicochemical properties like stability index, like what hydrophobicity, and again what is it called, solubility, and charged charged amino acids, and again it's molecular weight. Here the molecular weight, what we have seen about the Lipinski rule and all that doesn't come here. Okay, it is a biological compound that is chemically synthesized. Now again, this is another way of telling. So this is endoplasmic reticulum is the most important aspect for making a peptide made here, and it is being presented here. The protein going to the proteasome is shown. Okay. Now, so as I was indicating to you earlier, MSC is highly diversified, and your MSC, my MSC is not the same. But that is a different thing with regard to the blood group. You may be having a blood group, I may be having a blood group, both are same. My blood suits to you, your blood suits to me. Okay, in case of emergency, I can give to you or I can take from you. But While giving the blood to you, to me, RBC does not have MSC molecule. That's the most advantage for us. RBC is enucleated. There's no nucleus in RBC, so it does not have the MSC molecule. But if I give my blood to you, it is having what is it called WBCs. Okay, WBC number is far less compared to the RBC. But WBCs are having the MSC molecule. Though those MSC molecules may not be compatible to your system, they may not be compatible to your system. Then the longevity of our WBCs is 4 to 5 days, so that it cannot elicit immune response. Okay, so that is the advantage of blood transfusion. But with regard to the organ transplantation, organs are having MSC class 1. Okay, so that's what we have to take all precautions at the time of organ transplantation. So as I said, this individual is having MSC class 1A, class 1B, class 1C, class 2 DP, DQ, DR, DM. So like this, there are good many number of HLA molecules. One important thing I will let you know, which may not come under the vaccine development, you all as participants should get to know. There is HLA class 1 contains class 1A, class 1B, class 1C. There's no D here. E, F, G. I wanted to make you to get to know what is G. HLA G, when the fetus is there in the womb of the mother, the fetus is not rejected by the mother. Otherwise, the fetus contains half of the HLA molecules of the father. So those HLA molecules are different from mother, so that mother has to reject it. Okay, it doesn't do because in the fetus only HLA class 1G is expressed, not A, B, C, E, F. Only G molecule is expressed. G is least polymorphic. G is least polymorphic. As a result, it is being maintained in the womb, and so that after having taken birth, it expresses this HLA A, B, C. So that is the case with regard to the fetus HLA composition. Okay. Now these HLA molecules are being present in the genes of these HLA molecules are present on the 6th chromosome. We've got 23 pairs, 6th chromosome, and in the 6th chromosome, they are arranged in this way: A, B, C. That is HLA class 1A, HLA class 1B, HLA class 1C. They are arranged like this. It's going like that. Okay. So here we have got HLA class 2, DP, DQ, DR, DM, DP like that. Okay. So now we are not looking into HLA class 2 here. We are going through HLA class 1 because we are handling the viral proteins here. So in this case, let us see the polymorphism. Just now I gave you the example of ABO blood group. In the blood group, we have got only A, B, and not having the gene is O. Okay, like that. So in this case, if we take up, A is 559 alleles. B is around, A is around 303 alleles. B is around 559 alleles. And C is around 150 alleles. So this is the information I got it. But if you go through the latest information, they may vary more. Okay, so like this, that means MSC class 1A is having 559 alleles. What allele I'm having, you may not be having. And again, the combination, I may not have only MSC class 1A. I'm having class 1A, class 1B, class 1C, A, B, C. All three alleles are with me. I may be having B allele of 10, C allele of 60, A allele of 110. So these three alleles are there. This combination you may not have. No one will be having. So that's what called the haplotype. Haplotype. So that we have to work out. Okay. Thank you. Now let us see the focus on HLA polymorphism. A few human beings will share the same set of HLA. How is that? A few human beings share the same set of HLA alleles. That's what called consanguineous marriages. And again, the monozygotic twins will be having the same ditto, same monozygote, not dizygotic twins. Monozygotic twins will be having the same set of HLA alleles, and sharing of HLA alleles is possible in consanguineous marriages. So one more thing I want to let you know here, particularly in the zoo park, the tigers, lions, and all those things, They're all the progeny of one particular parents, one particular parents. So they all will be sharing the same HLA molecule. They're also having HLA MSC molecule. They share. As a result, they'll be having frequent, what is it called, the infections. And apart from that, their lifespan is short. Their lifespan is short. So that's what HLA molecules, as diverse as they are, as we see in the case of human beings, will be having longevity also in this life. So different persons will react to pathogen infection in a different manner. A T-cell based vaccine must include epitope specific for HLA allele in a population. Okay. So population-wise, the HLA alleles will vary. As a result, we also collected from Andhra population. What are the HLA prominent HLA alleles? Okay. This is another important point. A peptide-based vaccine. If you make a peptide-based vaccine, as we have got the recombinant vaccine that we are having for hepatitis B surface antigen, must consist of nearly 100 epitopes, which can bind to class 1. MSC 100 epitopes. Okay, with one epitope, we cannot make a recombinant vaccine. That's what called with 100 epitopes. If it is made, it's a polytope. It's a polytope. So, like that for bacterial, particularly for MSC class 2 epitopes, there will be more than 1,000. So, this is all the recombinant based vaccines wherein the epitopes are of this many number. Remember that. So, those are the participants who are planning to make a polytope. Okay, we have to remember that the epitope of this much of combination is required. So now again, there are good many databases like IEDP, Immune Epitope Database, CFITI, and Protein Database. They got so many of this HLA, what is it called, sequences with them, HLA-B. So if at all you want to work with IEDB software for the epitopes of your choice, it will show you HLA-34 HLA molecules, it will show you. HLA-B shows 28 like this. Okay. So now we are coming to the fag end of this MSC class 1. See, this is what the epitope, the binding or anchoring to the grooves or cleft of MSC class 1. So like that, this MSC class 1 coupled with the peptide is being accepted by the T helper cell. So this is what the structure shown along with the bioinformatic tools. So now, this is again another interesting for those of young participants who are not familiar with the specificity. Now, let us say we've got MSC molecule of this type, MSC class A. Okay, MSC class 1A, class 1B, something like that. Okay, so if the epitope, the protein is fragmented, the proteasome. Several fragments have come from the protein. Okay, all fragments are not binding to all MSC molecules. A few fragments, this fragment bound, anchored to the MSC molecule. Okay, so this MSC molecule through endoplasmic reticulum and all that, it takes to the cell surface. Like that, this MSC molecule to which the fragment is bound, it is anchored, it takes to the cell surface. Okay, so this is the most important aspect of immune system for antigen presentation. Because the antigen is being presented, the T cell is in a position to receive it. Otherwise, the T cell is ignorant. T cell is ignorant. It doesn't know what is this infection, what is the infection. Okay, the T cell has to be made interacted with this dendritic cells. Okay, so that's what here. So here our interest, our bioinformatics interest is these many peptides which are coming from the antigen in the process of the proteasome lysis. Not all peptides are epitopes. That's what we should get to know. Not all peptides are epitopes. A few which can anchor, which are having the physicochemical properties to bind to the MSC class 1, they become the epitopes. That's what I've been emphasizing. So that's what the bioinformatics tells us. Okay. Now, so that's what I told you about MSC class 1. Now let's see online software tools with which you may be familiar much. So this one slide only I kept it. Now we are all familiar through the NCBI, with which we can take the protein of our choice, or the antigen of our choice, genes of our choice. Okay. So then we can go to the PDB, where in the PDB files of the proteins because at times to submit to some of these online tools, we require PDB file of that protein. Okay, so from that we can procure from this. Okay, so ITAS, whatever the protein, whatever the epitope that you are making, whatever the polytope that you are designing, and that protein structure and protein modeling and validation of this model, all that has to be done through ITAS. Okay, and now this is the most important aspect, immune epitope database. And really, I got astonished once I presented a protein, a spike protein. Recently, I did that work. It has given so many in the Excel sheet, so many epitopes. What to select, what not to select. There's become a big confusion. So, like this immune epitope database, each of you work with the protein of your choice. Now I will be showing this spike protein. With that, all participants will work. We are not involved in what is called this demo session here. Okay. The next one is any polytope we make, and we had to get a sort of a certificate. It is free from allergen. It is non-allergic. It is non-allergen. Okay. So once you submit that polytope sequence to allertope 2 version 2, we'll be getting non-allergen, allergens. Okay. Like this, the polytope toxin peptides. See, some of the peptides are toxic. The snake venom contains peptides. Scorpion venom contains peptides. So like this, the peptide that you are making, the polypeptide that you are getting may be toxic. So for that, again, you have to submit to the toxin parade, wherein while you are submitting the toxin parade, you have to give less than 50 amino acids at a time. It doesn't accept all 108, 200 amino acids of your polypeptide. Okay, so then it gives whether it is non-toxin or toxic. Okay, so like this, there is another important thing called CIMSIM. That is computer immune simulation. Computer immune simulation. This is wonderful results we'll be getting, and for which you should get to know the depth of immunology, wherein once you give the vaccine, that is in silico vaccine, not real. So take a vaccine, prepare a epitope, and all this epitope prepare a sort of a paratope, polytope, and that polytope sequence if we give to the this computer immune simulation, then there are several parameters, whether it promotes the T cell, whether it promotes the B cell, whether it promotes the macrophages, okay, whether it promotes IgM, whether it promotes IgG, enhancing their levels, and again, does it promote the cytokines, whether it causes cytokine storm. Several of these parameters you can study through the computerized immune simulation. Like that, B cell epitope prediction, and again, the solubility of the epitopes, and several things, even cypress, all these things can work better. Youngsters, there's good chance for you to work. So this many software tools, once you get into that, you get coming across several like RankPep, that is epitope prediction through RankPep. So several other things are there. Even from our India also, IMTech, that is Institute of Microbial Technology, earlier there's Professor Raghava. He has developed good many techniques relating to this immunoinformatics. You can follow. Just if you type the Raghava, and it gives good many type of tools. Okay. Now, so the need for computer-aided design. So we have gone through what is the need for vaccine. We have gone through this brief outline of immune system, and we have gone through MSC1 complex. What is MSC? There is major histocompatibility complex 1. Later, we have gone through the software tools which are used for this immunoinformatics, and again for the development of the vaccine, in silico vaccine design. And now, need for computer-aided design. Why? For all the subjects, there is no computer being used at length. For this immunology and for vaccine, we are telling so much about the vaccine. Let us see the requirement. So the immune system is highly complex. There is, as I told you, a lot of diversity is there in MSC molecule. There's 10 raised to 13 MSC class 1 haplotypes in the population. So this is being shown in IMGT, that is immunogenetics database, and HLA data. There's a database on HLA, human leukocyte antigen. Okay, there's a database on immunogenetics, and that will be getting this information. 10 raised to 13 MSC class 1 molecule. Those were the days people were studying about the human migration from Africa to East Asia, and from there to South Asia, how the people have moved. They are now evaluating through the primers the MSc class 1 molecules. Through that, they are studying the human mutation. Now it is taken so much importance with regard to the vaccine development. Okay. Now, so there is a 10 to the power 15 different type of the T cell receptors. This is also a good amount of variability. And again, 10 to the power 12 B cell chronotypes. So many clones of B cells. That's what I have given one example of Burnett, McFarlane Burnett, about the B cell proliferation, clone of B cells. Okay, then 10 to the power 11 linear epitopes, composed of 9 amino acids, and 10 to the power 11 conformational epitopes. So this much of complexity is involved. As a result, we require computer-aided drug design and this vaccine design to have some sort of confirmation results. Okay. Now, so let us see here. So this is what our machine learning tools. We can devise the training, as we have understood now. Like you all, I also understood the machine learning. Of course, some of you must be experienced supervised learning, unsupervised learning, classification, regression, and reinforced learning. Okay, and all that training sets, and again the test sets, and again what is it called? The other aspects known as the node repository. So like this, if you give the information to the computer, the antigen, and it gives several of the epitopes. Okay, so one of these epitope is a peptide. That peptide binds. This is what I've been telling you earlier. Peptide binds the MSC. So this identifying this peptide is a Herculean task. Herculean task for a bioinformatician. So then if you identify this stuff with one peptide, we cannot make a vaccine, as I told you earlier. Then we should have more than 15, 20, or 100 peptides to become a polytope. So if it is so many peptides, means again synthesizing those peptides, it is a cost is involved. So this is what here designing this peptide. Okay, and again this peptide is nothing but an epitope. Epitope is the one. The epitope is the one, that peptide of this protein getting anchored to MSC is called epitope. That's all. Okay, it is antigenic determinant site. Okay, so this is what involved in this machine learning, particularly in this vaccine. So many combinations are there because there's 21 amino acids. If you are polar, if you are hydropathic, if you are hydrophilic, okay, if you are non-polar, something like that, and if you are aliphatic, and chain reactions, several reactions are there which we had stabilization, all that. Okay. Now, so identify epitopes in a plant in a genome. Identifying epitopes in a genome. Later, a small viral genome contains 1,000 potential. See, small viral genome, our COVID, let us say 1,000 potential cytotoxic T lymphocyte epitopes. Bacterial genome contains 1 lakh epitopes. Okay, no two humans will induce the same reaction to a pathogen. So that's what now. If I take Covishield as vaccine, I may not respond. But some of the others who take the Covishield as vaccine, they suffer for about 4 to 5 days. They have to be given the dose tablets. So that's what no two individuals react in a similar way for any pathogen or for any vaccine. Okay, viral escape and viral genome diversity has to be understood. Okay, so that's what now predicting peptide MSC interaction. And again, application to epitope identification and vaccine design. Application to epitope identification vaccine design is like finding the needle in the haystack. So that's what I told you in the beginning itself. We have shown one, what is it called, funnel, wherein the genome of a particular virus or bacteria gives so many potential, what is that called, epitopes, of which consensus epitopes, confirmed epitopes, virulent epitopes. This is what the procedure. This is like finding the needle in the haystack. So now there's all about the background. Let us see a sort of a experiment. There is a bioinformatics experiments done by our students in our university. And for our second year students, I'm teaching the immunoinformatics. They're all quite familiar with this. and a few of them must have been attending this conference now. Okay, so this is the, what is this called, the spike proteins of the coronavirus, and nearly 16 proteins have been identified in this, like this envelope proteins. Envelope protein is highly, what is it called, highly consensus sequences, but the spike protein is now varying a lot, and I will show you now. So like this, there are good many number of proteins. And see, the participants, most of you may be knowing this, but those who are not familiar, please get to know about this GIS8. GIS8. Okay, this is Global Initiative on Sharing All Influenza Data. Global Initiative on Sharing all influenza data. In this, you'll be getting the latest variants of coronavirus. Not only coronavirus, other viruses also. Coronavirus is because we are all now contemplating on this. We'll be getting the latest variations. Okay, so this I collected a little back. So, okay, you please go through. And as a result of this, we are going to have in this coronavirus, S clade, L clade, V clade, G clade, GR clade, GH clade, and other clades. India will be having a few of these clades, and still this number must have been more now, the latest information. It is a little bit around 5 to 6 months old information. Okay. Now, these are all the various proteins that are there in this spike, in this coronavirus. Around 12 proteins I collected from PDB database. And this is the genome from NCB I collected, nearly 30,000 nucleotides, wherein the spike one will be having approximately around 1200 amino acids. And now, so just as shown pictorially, the spike protein is having several epitopes. Of these epitopes, our concern is on receptor binding. Which of the epitope binds to the receptor? That is most important. So that is, we are having the now called, what is it called, angiotensin converting enzyme, ACE2 receptor, it binds. So that's what we are going to focus. So now let us see a brief about these several viruses. Wuhan coronavirus and Middle East respiratory syndrome, MERS, and severe acute respiratory syndrome virus, SARS, and common cold caused by coronavirus. Okay, so like this, there are 4 varieties. So in these 4 varieties, get to know this common cold caused by coronavirus. There are 4 types: 229E, NL63, OC43, HKU1. So these are all called alpha coronavirus. Alpha. These 3 come under the beta coronavirus. This beta coronavirus must have taken origin from the alpha coronaviruses. We also have done the phylogeny, which I did not show you here. So that's what this is alpha coronavirus. These alpha coronaviruses are there circulating throughout the year, but common in fall and winter. They are not what is it called, that much tragic. They do not cause that much toxicity or whatever case may be, virulent. They are not that much virulent. But these viruses have become virulent, and SARS was there in 2002, predominant in southern China, and it is declined now, 2004 onwards, it's not seen. But MERS also declined. It was linked only to the Arabian Peninsula, and now it is declining since 2016, mostly because of herd immunity, development of the herd immunity. But this Wuhan coronavirus is predominant. Its various viral escapes are predominant. It is spread across the continents and all over the world. This variants, viral escapes of coronavirus spreading. So this is something about the relation of our coronavirus to other coronaviruses, if you must be knowing. Now, so now let's go only to the spike protein. Though the spike protein of bad coronavirus ranges. The spike protein of it is in this range, and China SARS virus also will be having this range, and MERS Egypt, and this also varies in this range, and our new novel coronavirus is having. So that means this receptor binding protein of the spike protein is in the range of between 300 to 540, 550. So if all of these are getting anchored to angiotensin converting enzyme 2. Only this MERS is getting anchored to dipeptidyl peptidase enzyme receptor. Okay, so these alpha coronaviruses, as we have shown before, they are not getting anchored to this. That's what they were not serious in their virulence factors. Now, so we are now focusing in our further, this is all a case study project work that our students have done. We are focusing on RBD, receptor binding domain only. We are not taking all 1200 amino acid sequences of this, only receptor binding domain alone we have taken for our experimental purpose. Okay. So that's what once we are given and we submitted to IEDB software tools. This is what our Andhra Pradesh haplotype, HLA haplotype. So in this HLA haplotype, we have got HLA A01, HLA B57, and HLA A33, and HLA A02, and HLA A21, and B07, and HLA A24, B40. Okay, so the others one what we are showing here, DRB1, and DRB1, and DRB1, and DRB115. They are all called, what is it called? The HLA class 2. Okay, so now for this, against this HLA, this many epitopes have been identified. Okay, so several have come, of which a top 10 they have rank of 100 they were selected. Okay, now this we have taken to this ITASR, and wherein the secondary structure of this polytope. From the epitope, all these 10 epitopes have been fused together with certain amino acids, so that in the system, once the polytope goes in our system, it needs to be cleaved by proteolytic enzymes. So as a result, we have to insert certain amino acids, linking these epitopes. Okay, so such a type of the design, the protein polytope, what we designed is nearly 108 residues that we submitted to the ITASR. It has given this homology modeling like this, and here more of beta Peter sheets, and coil, random coil. There's one alpha helix. Okay, so for this we have taken the ProCheck. What is this called? This Ramchandran plot. Here the validation of the structure, where this beta Peter sheets are shown here, and this alpha helix structure only one is shown here. So like this homology modeling and validation was done for the polytope that we designed. That's what I'm telling you, the participants. If you want to design any vaccine, first to go to the epitope prediction through the IEDB tools. Later, you go to the later you have to couple all these epitopes, the best epitopes that having the virulent factors with the amino acids of your choice, so that they'll be broken down, hydrolytic breaking down take place in the proteolysis. Then we'll be going to the ITASR, developing the model, and again studying its validation. Then in this, we have to study the what is it called, discontinuous epitopes or conformational epitopes. So that 108 that's the residues of this polytope. Once we submit it to this IEDB, then there's several sub protocols in IEDB wherein it has given this conformational epitopes, this many, number 8, and again it gives the view. Of course, it doesn't give here the binding energies, it gives the view where these conformation epitopes are getting anchored to the antibody. Okay. Now, so as I told you that in the softwares which I have shown you, we have to take almost like a certificate. The polytope that you made, is it toxic or non-toxic? So like this, we had this 108 amino acids we have made in our polytope, wherein we cannot submit to 108 as it is. Less than 50 amino acids have to be given. Once it is given, it is shown this much of score: molecular weight, charge, hydrophilicity, and hydrophobicity, and prediction. So here, this is most important part. This peptide is non-toxin, and again subsequent peptide is non-toxin. Again, subsequent peptide is non-toxin. Put together, all this is 108 residues which constitute the polytope of our design, what we designed here from the spike protein of coronavirus. Okay, now like this, if you submit to the alert top also, because ultimately we are end users, whatever the software that are developed, we use them and present in compliance with the concepts that are developed. So here also it gives probable non-allergen, whatever the one that we submitted, polytope, for which it has given the response, alert top as probable non. This information is required for any publication, for any production. Okay, now this is what I was telling to you, CIM-SIM, that is computer immunological simulation. So that peptide of 108, if you're given, what are the active T helper cells? Okay, what are the active T helper cells? So once it is given, the active T helper cell proportion is increased in days. Later, in so many days, after 100 days, the proportion got decreased. Okay, so that means they're all becoming dormant. Once there is sort of an activeness, arousal, then all this become now. So that's what the polytope, once we submit to the CIM-SIM, then it gives this polytope will promote so much of production of T helper cells. Okay. The next one is this polytope also gives the B memory cells. The B memory cells, and these B memory cells will be there starting from 25 days and then 50 days. Then it will be there till 100, 150, 200 days. That's what the people say. Those who are relaxed, got relief cured from this coronavirus after 3 months. Also, we can collect their plasma because they got this many number of the B cells with the immunoglobulins, that their plasma can be used for therapy. Okay, so not soon after their recovery, after 100 days or 250, 150th day of the recovery, you can take their plasma. Not after 300 days of recovery, the plasma cells and immunoglobulins will be lessened. So this is again with regard to the immunoglobulin M and immunoglobulin G. So we used to say, so that's what whenever this polytope is given to begin with, see here, blue one, this is what the IgM will come. That's what the rapid test that is there in vogue for this coronavirus. There is an indication of IgM, there is an indication of IgG. It shows both. If IgM and IgG both are shown, then it is positive. If IgG alone is shown, it is not positive because we've got IgGs for several of our systems in our system. Okay, if IgM and IgG both are shown, there is an indication that we are infected. So that's if the polytope is given, it gives an indication of what is the IgM levels, what is the IgG levels, predicted IgM. Okay, this is all computerized simulation. Then this is another interesting feature. So soon after taking vaccine, a few people feel a sort of body ache and fever, inflammation. Okay, it may not be there for all. This is what it is not infected. It is not because of the infection. So while taking injection, people say I got infected. It is not infection. It is because of the vaccine that's making the cytokines to get released. And these cytokines disturb in such a way that there is a commotion, there is a confusion. As a result, we are suffering for about a day or two, and it will become normal. It is called as this cytokine storm. Okay, these many things one has to do if an epitope is designed, polytope is designed for any protein of your choice. Now, what is happening is only for 30 diseases the vaccines are through. Still, there's several of the diseases participants can plan now with all this immunosomatic dose. So, advantage of epitope vaccine is more specific immunity, devoid of side effects, the ability to deliver high doses of potential immunogen, and it is worldwide conservancy of epitopes. Again, stable binding with HLA class 1 and smooth passage through transport-aided protein. So with this, I'm coming to the fact end, and here the summary of this is the need for the computer-aided design was elaborated for the past 1 hour or so. And hereby we acknowledge all the sources that were used from the net for developing this workshop material and teaching material. And I inform to all the participants, others, don't do this type of coughing or sneezing. It's so much of what they call vapor from our mouth or nose, so that it carries what are the germs that are there with you. Always, this is a ill health practice. See that you wear the mask and save your life and also to save the neighbor's life. Okay. Thank you, one and all. Thank you. So
Participants can unmute and ask questions.
Yeah, I'm happy to respond a few questions.
Yes, that was a fantastic session. Participants can ask the questions. Yes, Sivaraman is saying that is very informative. Thanks for the lecture. Thank you, Sivaraman sir.
Thank you. Any questions? I'll be happy to hear a few questions now.
This is Dr. Shivaraman here. The session is very informative. Thank you so much, sir, for your time and pain in preparing this presentation. Sir, like I have one simple question. So you have detailed a bit about like framing the epitopes and identification with the help of some software tools being available. Sir, like if probably could be in vaccination, we should have a task of designing the memetopes that complement to these epitopes, no, sir? Could you please brief the audience about the process of framing or designing these memetopes? So it could be highly helpful for me and even for my colleagues even.
Thank you. I am familiar so far about the epitopes and also the polytope. Memetopes I have not gone through. I may not be in a position to let you know. So once I go through the details, I will let you know later. Okay, I will make a note of your email from Abraham and I'll clear it to you later. Okay.
Thank you, sir. Thank you so much.
Thank you, Krupanidhi sir. It is saying that it is very interesting.
Any more questions? I can also learn from the participants. You can ask a few more.
Excuse me, sir. Am I audible?
Yeah, audible, sir.
Yeah, sir, I am Abhijit, assistant professor at NIT Pharmacy Institute, Greater Noida. Sir, I have actually not question. I just want to know from you about the future of vaccine, because sir, nowadays mRNA vaccines are top US-based companies are promoting mRNA vaccine. So, sir, what is your opinion on what will be your suggestion to us about the future of vaccine?
Yeah, what is happening? Yeah, many good question about because we're all now hearing in the news and mRNA vaccine and vectored vaccine, mRNA vaccines from this Moderna and Pfizer, and vectored vaccines from our Serum Institute and also from Gamaleya, Russia, have come out in the market. But still the recombinant protein vaccine has not come, like hepatitis surface antigen, hepatitis B surface antigen. But all over America, USA, the mRNA vaccine is given. They have not imported our from India, what is it called, Covaxin and all those things. Because let us see how best they are getting immunized, because it is the predominance of the industries, they are dictating what to follow in the society. But out of all, the inactivated vaccine that is coming from Sinobiotech from China and Bharat Biotech from India are the best for any type of variants that are coming in the pathogens. But to some extent, the recombinant antigen is also good. But now the vectored vaccine has come and the mRNA vaccine I do not think so they will last long. Okay, it is my prediction, but it may not be correct because companies are investing billions of rupees on that. Okay, just my opinion.
Thank you, sir. Thank you for sharing this. Thank you, sir.
Any more? Thank you, Abraham, for the nice workshop that you are organizing. Thank you, Hima.
Yes, sir. Sir, I would like to bring you on kind notice that our second year bioinformatics students are following the R19 curriculum, in which they carry out some kind of project work, minor project work, along with their subject. Immunoinformatics is one of the 6 subjects, and as Professor Krupanidhi sir said, it is being taught by him. With all your permission, I request one of our BTech second year bioinformatics students by name Venkat Chaturya to present her minor project, sir, for 5 to 10 minutes.
Okay,
she has carried out in this lockdown period via online classes. After that, we'll have a valedictory session. It will get finished in 15 to 20 minutes. Not a problem, sir. Can I ask Chaturya to share the screen? Kupan is if you stop screen sharing, you will see.
Stop sharing. How to stop it? Is it stopped?
Yes, it's gone.
Very good.
Chadira, can you share the screen?
Yes, sir. 1 minute.
Yeah. Where are we? Yes, we're able to see your screen. Please be quick and finish within 5 to 7 minutes, Chadria. We'll have very little time. Go ahead.
Yes, sir. So, good evening, everyone. Nowadays, we hear this word every day in this pandemic, and everyone are eagerly waiting for that, and that is vaccine. Everyone are trying really hard to get vaccinated nowadays. So, have you ever wondered how is this vaccine created or prepared? I and my friends tried to prepare computational vaccine using bioinformatic tools. First of all, let us know what is a vaccine. Vaccine is a biological preparation that provides active acquired immunity to a particular disease. It is often created by a whole attenuated organism, microorganism, or its surface proteins, or its toxic peptides, etc. Vaccine was discovered by Edward Jenner against smallpox virus using cowpox virus. And the word vaccine was given by Louis Pasteur. And this is the history of vaccine. There are six, in classification of vaccine, there are 6 types of vaccines. That is whole inactivated vaccine, live attenuated vaccine, DNA vaccine, purified macromolecule vaccine, subunit vaccine, and recombinant vaccine. For this project, we are dealing with multivalent subunit vaccine. Multivalent subunit vaccine is a vaccine which is designed to immunize ourselves from 2 or more strains of the same microorganism or more microorganisms. This is a synthetic peptide vaccine. Examples of this is hepatitis B, tobacco mosaic virus, and coronavirus. Multivalent subunit vaccine for coronavirus is yet to be discovered. Steps for vaccine development. In vaccine development, first the microorganism is selected. We will grow this in labs, and this is isolated, purified, and inactivated, and forms the vaccine, and they are packaged and administered to us. Mostly this is the conventional method or the traditional method of preparing vaccine. It takes more time and also cost effective. Therefore, we can prepare vaccine beforehand, and that is beforehand using computational methods, and the term is called reverse vaccinology. The word reverse vaccinology means reparing a vaccine using computational servers or bioinformatic tools. The word reverse refers to the used or expressed DNA over purified proteins from the organism itself. So the best example of vaccine created by using this approach is meningococcal B vaccine against Neisseria meningitis. This procedure was most helpful in developing subunit vaccine. The advantage of preparing a vaccine using this approach is that it is cost effective, and also it takes less time to prepare. Steps to be approached in reverse vaccinology is same as the conventional methods or the traditional methods, but except that we use informatic tools here. So designing a vaccine using bioinformatic tools. So for this, we selected hepatitis B virus to design a vaccine. Before going to creation of that vaccine, let me tell you about hepatitis B. Hepatitis B disease. Hepatitis B is an inflammatory condition of liver. It is commonly caused by viral infection. It approximately 4.4 million Americans are living with chronic hepatitis B and C. They are classified into 5 strains: A, B, C, D, and E. B, C, D are long-term and ongoing virus which causes a chronic disease. A and E are acute. Causes of hepatitis B are excessive alcohol intake, excessive dosage of medication, exposure to poison, etc. Steps in vaccine design. First, we need to retrieve a protein sequence, and we have done this using NCBIs and PDB servers. So this is the FASTA sequence of hepatitis B virus we took for the vaccine construct. And this we need before taking this into consideration, we need to also check whether it is antigenic or not in order to produce the immune response in our body. And we have checked that in VIGNAN'S server. So this is VIGNAN'S server where we can check the protein sequence or the genomic sequence which we take is antigenic or not. So here it tells it is. So here it tells the result of it. This is the result of the protein we took. It says antigenic. And next is selection of CTL epitopes. The selection of CTL epitopes were done in server using NetCTL version 1.2. This is NetCTL 1.2 server where we have got the CTL epitopes for the protein sequence we took. Not only the CTL epitopes, but also we need to check the antigenicity, allergic properties, and toxic properties of the epitopes we'll be taking. So these two are the epitopes we took for the vaccine construct. And the E here tells us that it can produce immune response. And these are the best epitopes to carry forward. And to check the, as I said before, to check the allergic properties, we check this in alert spread. We can also check this in alert top, as sir told us before PPT. This is alert top server, but we did it in alert spread, and this is the result of that. It says non-allergic. And next is toxicity of the epitopes. Even it plays a major role in preparing it. And this also gave us that it is non-toxic. Next, we've checked the immunogenicity of the epitopes. As you can see here, the score you can see here. The higher the score, the higher it gives the immune response in our body. These are the CTL epitopes we use and the score of it. The next one is a prediction of HTL epitopes. HTL epitopes are, we predicted HTL epitopes using IEDB server. This is the server where we have brought the HTL epitopes using the sequence. And this is the result of that, result of the sequence. And among these, we have took 5 epitopes, 5 HTL epitopes based on the by checking the conditions like allergic property, antigenicity, and toxic properties. We have used the same servers like Vaxigen, AlertSpread, and ToxinPred to check them, and the result output was like antigenic, non-allergic, and non-toxic. And we have selected 5 HTL epitopes among them. These are the 5 HTL epitopes we have took, and this is the ToxinPred server where it tells it's toxic or not. And here you can see that the all epitopes we took are non-toxic, and we took 5 which can fulfill the conditions like non-allergic and antigenic. And then in the next step, we have gathered HTL and CTL epitopes from the above process, and brought them together. And we also need adjuvant for vaccine construct, as adjuvants are used or added to vaccine in order to stimulate or produce antibodies against vaccine to make it effective. So the adjuvant we use here is HBCAG from NCBI. So the first 3 lines in this picture are the FASTA sequence of our adjuvant, and the next 2 here are the CTL epitopes, and the 5 here are the HTL epitopes. And we need to and we prepared peptide sequence using adjuvant, CTL, and HTL epitopes. And to prepare that, we need to link all these 3, and we are linking them. We link them using linkers. For adjuvant, we used EAAK linker, and for CTL epitopes, we used AAY linker, and for HTL epitopes, GPG linker. And we also checked the antigen property and allergic property of the vaccine construct prepared before, as shown in the before slide. And it says it's antigen, and the results were like antigenic and non-allergic. And also we have checked in ToxinPred, it says non-toxic. And we also need to know the chemical structure of the vaccine construct we created. So we have checked it in XPASI server. So this is the XPASI server where we have checked by giving our sequence or the vaccine construct we created. It gives us the chemical structure and its parameters of our sequence. And this is the says how many number of amino acids we have, what is its charge, is it stable or not, what is its half-life, etc. in this server. And next we have predicted the secondary structure of the vaccine construct or the amino acid sequence we created in PISPRED. It can also be done in JPRED server. This is the PISPRED server where I've pasted my sequence here to in order to know the secondary structure of our protein vaccine construct. And this is the result of it. Here, the pink colored boxes tells us they are helix, and the yellow colored ones are strands, and the gray colored ones are coils. This makes our secondary protein structure. And next, we also predicted the protein structure using ITASA server. ITASA server shows the 3D model of the protein. And this is a ITASA server. And the result of it was this. We've got 5 sequences. Sorry, 5 protein models. And in this 5 protein models, we have took the first one because in this, the C-score you see here, it is very important to predict a perfect or the desired protein. Favorably, C-score must be between -5 to 2. So the highest C-score I've got is -3.64. So I took this model for my further. Sorry, we took this for further thing. And next we've done the patch doc. And this is the patch doc server. Patch doc is used. Patch doc is something compared with a zigzag puzzle. As when we play zigzag puzzle, we compare the puzzle pieces whether it matches or not. In the same way, patch doc compares the protein and the compares the 2 proteins and gives the result whether it matches or not. So here we have took the TLR3 of human and the vaccine construct we've created. And this is the docking image of the vaccine construct and TLR3. Here you can see. And next, we also predicted the protein peptide interactions using LickPlus server. LickPlus server is a computer program that generates schematic 2D representation of a protein ligand complex. So this was the result of it. And in this, you can see it is divided into 2 lines, divided by a line. The purple colored ones is the TLR of a human, and the red colored one here is the vaccine construct we created. You can see the bonds between here. This is the protein and peptide interaction. And next is the Ramchandran plot for the vaccine construct we have created. According to this plot, the brown colored area is permitted area. The yellow colored one is partially permitted, and the skin colored one is not permitted area. All this red and blue colored dots you see are the amino acids residues of the vaccine construct we've created. So all are majorly in the brown colored area. So the vaccine construct we have created is permitted to form a vaccine. And these are the servers we use for our vaccine construct: NCBI, Vaccine, AlertSpread, ToxicRed, XFACI, ITASR, PatchDoc, and LickPlus, and also PDB-Sum. Thank you. Thank you.
It's an excellent presentation, Chatira. Any would like to comment on that? Any participants? Thank you for your great presentation. Any participant would like to comment on this? You can unmute yourself and make it.
So
Yeah,
Very, very nice presentation. Like, hopefully the people really make use of this pandemic time and they have come up with this beautiful module. Being as a student's project, it was quite amazed. And you please continue this research and look after this possibility of tagging with some suitable R&D of going into developing this peptide in vitro module, so that probably you can have a potential of kickstart of an initial phase of vaccine development. That was a nice presentation and it was beautifully made. And thanks to you. You should convey your sincere thanks to your mentor who guided you and framed you in a proper module of designing this vaccine. It was such a wonderful experience of hearing from this platform. Thank you.
That's so kind of you.
Thank you, sir.
This kind of support and encouragement, if these young students can get from you people, it means a lot to them to enhance their skills. For that, I thank you and all of my participants wholeheartedly. Thank you.
Thank you.
I ask Krupanidhi sir also to comment on this, and we'll further move to the valedictory session within few minutes. Yeah, Krupanidhi sir.
So I'm quite happy to hear from Chaturya, Monica, and others that they made a wonderful project work. And because I've been teaching this for the past 4 months to these students, they come out with good software tool acquaintances and also how to prepare a vaccine formula with adjuvant, with other aspects. And it is immensely happy that the students have grown very well. And this still you see that you improve by working on this. And you will be required by several industries who are in this line. And with the participants whomsoever are there with us, we are quite happy to have our students to get associated with you for future any internship at your program, and you people coming to our place and all those things. And many good appreciations to our students. Thank you one and all.
Thank you, sir.
Thank you, sir. Now, welcome to the valedictory session. Now, I would like to request Ms. Gayatri, second year BTech Bioinformatics student, to sing a song to start the valedictory session.
Classicals. Yeah, Gayatri, are you with us?
Yes, sir.
Yes, ma.
Yeah, thank you, everyone. Krishnani Megane Maro Krishnani Megane Maro Krishnani Megane Maro Megane
गाना सुनेगा है ना?
Sorry, ma, you can start again. Somebody has unmuted their mic, so you can start freshly, ma. Sorry for that. Some participant has unmuted their mic, so that I have to mute it.
Yes.
Actually, please start from starting.
Yeah.
Krishnani Megane Maro Krishnani Megane Maro Krishnani Megane Maro Megane Baro Mukhavan
तोरो बेगने मारो मुखवन्ने तोरो बेगने मारो मुखवन्ने तोरो कृष्णानी बेगने मारो कालानंदिगे जे घलु खल्लो यन्नुता कालानंदिगे जे घलु खल्लो यन्नुता नीलवर्णने नाट्य मारुत मारो नीलवर्णने नाट्य मारुत मारो कृष्णानी बेगने मारो काशी पीतांबर कैयल्ली ಕೊಳ್ಳಲು ಕಾಶಿ ಪೀತಾಂಬರ ಕೈಯಲ್ಲಿ ಕೊಳ್ಳಲು ಪೂಸಿದ ಶ್ರೀಗಂಧ ಮೈಯಲ್ಲ ಚಂದ ಪೂಸಿದ ಶ್ರೀಗಂಧ ಮೈಯಲ್ಲ ಚಂದ ಕೃಷ್ಣಾನಿ ಬೇಗನೆ ಮಾರೋ ತಾಯಿಗೆ ಬಾಯಲ್ಲಿ ಜಗವನ್ನೇ ತೋರಿದ ತಾಯಿಗೆ ಬಾಯಲ್ಲಿ ಜಗವನ್ನೇ ತೋರಿದ ಜಗತ್ತೋದ್ಧಾರಕ ನಮ್ಮ ಉಡುಪಿ ಶ್ರೀ ಕೃಷ್ಣ ಜಗತ್ತೋದ್ಧಾರಕ ನಮ್ಮ ಉಡುಪಿ ಶ್ರೀ ಕೃಷ್ಣ ಕೃಷ್ಣಾನಿ ಬೇಗನೆ ಮಾರೋ ಕೃಷ್ಣಾನಿ ಬೇಗನೆ ಮಾರೋ. Thank you.
Excellent. The
Thank you,
classical is
Gayatri.
always classical because it is classical.
Thank you,
Your,
sir.
yeah, your voice is so magical, ma. Thanks for enlightening us. Yes, Himashma, you may go with
Yes, sir.
reading about our department.
Okay, sir. Thank you, everyone. Now I'm going to add a few lines about Vignan and also about the Department of Biotechnology. The university was established in the year 2009 and it is accredited by NAC as A. The university gives top priority to academic excellence with social responsibility. This has been made possible under the dynamic leadership of Honorable Chairman Dr. Lau Rattaya and Vice Chairman Sri Lau Sri Krishna Devarayulu, who is a member of Parliament. The university is driven by values, ethics, and dedicated team of professionals with a great emphasis on innovation, research, skill development, and entrepreneurship. The university has entered an exciting phase and is shouldering greater responsibilities in imparting technical skills development in keeping with our Honorable Prime Minister's vision of Skill India, Make in India, and Digital India. Now, about the Department of Biotechnology. The Department of Biotechnology at VFSTR was established in the year of 2006. It was accredited by NBA in the year 2020. There are 25 doctors among its faculty members. It supports the extension activities such as rural development by promoting the cultivation of medical plants to the local farmers, especially among women. The department is very well established with COE animal cell culture facility, bioprocessing lab, plant tissue culture lab, bioinformatics lab, animal house, clean room, and biogas plant with an ample strength of students, 600, and faculty. The department has been supported by VFSTR, DST, and FIST and has attracted more than 10 external research projects to the tune of 33.5 crores. The department offers BTech, Biotechnology and Bioinformatics, BTech Honors, MTech, and also PhD programs, research-oriented programs. So now both BTech and MTech students undergo industrial internships and either in India or abroad. Every year, 20% of the students go for higher studies abroad, while 10% of students prefer higher education in India, and the remaining 60% students get placements in various streams. Thank you, everyone.
Thank you, Mashishma. Now, I may call upon Dr. Kalyani Dusya. Are you with us, Kalyani Dusya, ma'am? I don't see the screen.
Yes, Abraham, I'm here.
Yeah. Sorry, I don't see the screen. Yeah, you can go ahead. Please give some.
Thank you, Abraham.
Yeah.
Thank you. Welcome, everybody. And I hope this 2 weeks of workshop about introductory session for machine learning and a little bit of drug designing may have been an eye-opener and a stepping stone for all of our participants to build a career and look into machine learning as a venture to do more research. I know we all come from biology backgrounds, or sometimes we are very skeptic towards, oh, this is too tough, this is very mathematical, this requires a lot of computing, this requires a lot of coding. So all these stigma kind of hold all the students or earlier stage career research students to not go into these defining fields. But to all my students here, I would like to tell you that this is something you may want to invest your time into, because this will be very rewarding. As some of our speakers as well told you that text mining, machine learning, and artificial intelligence is the new requirement. Those of you who are looking forward to join research labs abroad, or maybe you want to go into industry, or somehow start your own diagnostic solution industries or something, this is where you have to look into, because there are a lot of jobs abroad, there are a lot of requirements abroad, and they don't have equipped people. Now, along with this, I would also like to add to this thing that I know in education in India is about, you get into the degree course and then you learn something. On the other hand, when you're going abroad or even for a job, now, India is changing very fast. You have to first prove yourself before you get into the job. So don't sit back and wait, oh, I will join this master's course in data science and then I will learn and then I will do project and then I will get wherever I have to go. That's not how the cycle works. This is the obsolete way of doing things, and you may fall in the run if you follow this procedure. So start doing things by your own today, and there is a lot of help on internet with digital media. There is a lot of help. All the workshop tutors that have given you instructions in past 2 weeks, they have good relations with us. You can always email them, and as students you have an advantage that it's called we have this scientific foundation where we have to help you. So if you write to any of the teachers, whether it's in your university or abroad or even in other universities, other states, they are bound to reply to you. And if they are not replying, that means there is something wrong with your email. So check that. If you keep on doing things in a proper channel, and if you channelize your energy, you will definitely get where you want to go. So this is some of the advice I have for all my students and other participants who are looking for a career in machine learning, or in who are looking forward to thrive in this industry. Other than that, I would thank Abraham so much for organizing this, and Vignan University University specifically for giving us all the permission for arranging this, and it has been so supportive. Thank you for all my tutors who have given presentations in past 2 weeks. You all have been great, and it's a wholeheartedly thanks to all the participants who have been here. You have been an amazing audience. All our emails are on the website. The YouTube links are there. Please, please, please go through that and contact us. We would love to hear from you. And with this, I would conclude my remarks. So Abraham could take over. Thank you, everybody.
Thank you, ma'am. Now I would like to call anyone to share their feedback. So Shivaraman sir, can you share your feedback? Or anyone else can share their feedback.
Hello.
Hello.
Would anyone like to share their feedback on the session? Please, they can.
Yes, participants like Bharat, Swapna Vadlamani. Please share yours. Karan, Afreen Khan. So many interested participants are there.
Shivaraman sir.
Yes.
Anyone can.
I cannot see their names. Yeah, Aditi Baduria, Ajit Singh, Anil Panwar, Aisha Siddika, Jitesh Kumar sir.
Excuse me, sir. Can I share?
Yes.
Yeah, my name is Abhijit. I am from NIT Pharmacy Institute in Greater Noida. So it is a great time with you all and all our very qualified and very knowledgeable our instructors. So being a teacher, being an assistant professor in pharmacy, I have learned a lot. And the world is now shifting to artificial intelligence, machine learning, and deep learning. So whatever the techniques, the skills that we have learned, so we're going to use those things in our upcoming teaching and in our upcoming research activities. So this is just the beginning for people like me. So I'm deeply thankful to you, sir, and your organization for organizing such a very interesting event. Earlier time, I was used to search on YouTube, ResearchGate, and other various platforms, but these things are not in very organized, and things are very scattered. So now, after learning from directly and a very organized and a very perfect manner, now the things are so much clear for us. So I'm deeply thankful to you, sir, for organizing this event, and I hope this will be create a very great futuristic step for a whole over country also, because we are last year we have seen the drug discovery hackathon, and this year we have seen the event organized by you. So yeah, I'm deeply thankful to your organization and all the coordinators, and also the instructors for teaching us in a very nice, interesting manner. Thank you, sir.
Oh, wow, that's very heart-touching words, sir. Yeah, thank you for that. Thank you for such a nice feedback. Yes, Shivaraman sir, you can.
I'm sorry, I couldn't be able to unmute for some period of time. So in a nutshell, if I want to describe about this workshop, this is one such typical workshop that follow the pattern of the real hands-on workshop. So probably like starting from step by step initially, like starting from the installation procedures and how to go about uploading and how to find out those errors, and with respect to rectification, all the speakers are really humble enough in finding out or in guiding us in each and individual step where the hurdles is come up with. So like at this junction, like I would like to convey my heartfelt thanks to the organizers for framing such a wonderful workshop, and also like it will mutually benefit not only the masters or the teachers, so probably the students, those who are upon to take up the research project forthcoming or in the soon after or later in the academic years. Definitely here it comes to the role of an artificial intelligence or a machine learning tools, some way or other, either in the way of drug discovery or else in the way of an vaccine development. Either way it comes, like definitely it is going to get implemented. Probably the outcome will be measurable in coming up days in the form of some research papers or some presentation in the forums. So like this is a real some what you can say in open-heartedly I can say like a real workshop. That means a real practical hands-on workshop, beautifully designed, and good cooperation from even the fellow participants, because sometimes it suits with the Western timings. So all the time they are patient enough in learning to them, because sometimes it even going stretching beyond the limits also. They maintain a good, polite, very decent and professional crowd. Thanks for the organizers, and thanks for my fellow participants also, and thanks for the nice workshop.
Oh,
Thanks for the opportunity.
Thanks for you for touching all the aspects. Your feedback is artificial intelligently recorded, sir, for future use. Thank you, Dr. Shivaraman Dhanashekhar, sir.
Thank you so much, sir.
Now, Swapna.
Yeah. Sir, good afternoon, sir. When I first saw the workshop, I thought it's just a mixture of most guest lectures and very little hands-on sessions. But I think you have connected the dots, getting all the eminent speakers, and a lot of hands-on sessions were given, which were very, very useful to us. And I didn't even try to miss one session also. All the sessions were very interesting, and I think I just learned as a course of machine learning before through Coursera and all, but this workshop helped me a lot to have a very good idea and hands-on with the machine learning in drug discovery. And I'm really thankful for the organization and for organizing such a wonderful workshop. And I'm also looking in future also to organize such good workshops for the benefit of audience like us. Thank you very much.
For sure, ma'am. Thank you for your support. Oh, if no further audience would like to share, can I call Dr. Krupanidhi, Professor Krupanidhi, sir? Yeah. Yes, sir. Yours is followed by Deputy HOD.
Yeah. So the organizing secretary and everything for this workshop, Dr. Abraham, and experts and dear participants and students, all these 14-15 days along with you, I also spent good amount of time. I learned several aspects of machine learning, though I have been familiar with artificial intelligence for a long time, because one of my friends was an expert in that, but never I incorporated that in relation to biology or chemistry or pharmacology. Now with this workshop, attending patiently, and fortunately, because this curfew period, we are at home, we are not in the college. If we are there in the college, several other distractions will be there, administrative distractions. Now we are in the house, and we are attending our classwork and attending this workshop very conveniently, and we are immensely happy. And there are good many speakers. By name, it is better to acknowledge them. We had Dr. Rohit, we had Dr. Krishnendu Bera, we had Dr. Chenan from Japan, we had Prateek from Chatterjee from South Korea, we are having Kalyani from New York, and we've got Saad Khan, and we've got Jatish Joshi from Pune, and we've got Girnath Pillai and Kalpana Raja, and we had Nag from Kansas, we had Estella from Europe, we had Fuzi from Japan. Excellent talk. And at the end, we had Dr. Shyam Perugu. Okay, with all those talk at the end, having Shyam Perugu and giving the live examples from the plant sources, how to give the training with the test images, how to give all those information so as to have conclusion about what is the this particular plant, what is the name of the plant. So like this excellent on this mission learning we all have gone through. It's a good information for every one of us who are participating. And from here we can start as a group having Abram as a nodal point. We can have a sort of an association, mission learning in biotechnology and pharmacy or chemistry, something like that. You can coin and inform to him. You can have some sort of, as Fizi was mentioning, a sort of here we can have a voluntary association so that we will interact each other. We can have communication. There's no personal benefits here. It's only subject-oriented aspects we can do. We can have Vignan at the center. Our management all through, what is it called, encourages this. So I want a few of seniors who can orient well, who can mold well, and you can join with our Abraham and see that you can say all of us are new in this. We are not experts in the machine learning. We are not experts in artificial intelligence. And new people can venture into a new aspect so that we can come out with the best solution. So it's better, Dr. Abraham, to have a few of the experts among the participants and also experts like Kalyani Madam, others, and now to join in this sort of an association with regard to the machine learning in biotechnology or something like that, whatever the name that we coin, so that we can grow together perpetually forever, and we can make our students to train in this type, particularly as Madam was mentioning, the text mining is having good, what is it called, benefits and career opportunities in USA and Australia, so that your students from your college and our students from our college, both of them work together so that they all will meet and our horizon will enhance. Okay, thank you one and all for participating, and it is a wonderful session. Really enjoyed. Abraham has given us good homework for all of us to do in this machine learning. Thank you one and all. Thank you. Abraham, sir,
Thank you, sir. Now, I would like to welcome Co-Convener Deputy Head Biotech Professor Venkateshwarlu, sir, to give his remarks on workshop.
Hello. Vote of thanks. Yes, yes.
Yeah. Hello. Good evening, Abraham.
Good evening, Professor Venkateshwarlu, sir.
Good evening to one and all. So it's a great honor and privilege to propose the vote of thanks on the occasion of the valedictory function of International E-Workshop on Machine Learning Applications in Drug Discovery, Basic to Advanced, organized by the Department of Biotechnology, VIGNAN'S Foundation for Science, Technology and Research. So I would like to express my heartfelt thanks to all the keynote speakers of this International E-Workshop. So the keynote speakers, namely Dr. Holong from Kansas State University, USA, Dr. Chanin from Mahidol University, Thailand, and Dr. Fuji from Astellas Pharma, Japan, and Dr. Kalyani from Albert Einstein College of Medicine, USA, for their lectures and training sessions in this workshop. So I want to extend a special thanks to all the invited speakers for sharing their valuable knowledge and training to the participants. So in 2 weeks workshop, so the experts emphasize on machine learning applications. So I will illustrate a few here. So the Mr. Rohit Sukla and Krishnan Bera and Chatterjee team has shown us the usage of different tools such as the modeler, IDOC, Pyrex, and simulation tool Gromax also. Dr. Kalyani trained the participants related to the decision trees and random forest applications in bioinformatics, and Mr. Saad Khan showed the the participants about the Python-based code to do the secondary structure prediction, and Jitesh Joshi gave extensive training on the vector tools to show the classification and regression models related to bioinformatics with a diabetes case study. So Dr. Girinath Pillai made the participants to work on the tools like NIME and Orange softwares for drug design. So Dr. Kalpana Raja, Dr. Pelugu Sham, and Kobe Panford were emphasized on the use of TensorFlow with the Google Colab Network, and Ms. Estela has talked about the case study of artificial intelligence applications in biomedical technologies, and Dr. Fuzi has introduced the small molecule database and predicted the drug likeness of small molecules using RDKT in Python network notebook. So today, our internal expert, our head of the Department of Biotechnology, VIGNAN'S Foundation for Science, Technology and Research, so Professor S. Krupanidhi, gave insights on various aspects of immunoinformatics in design of vaccine. So I express a special thanks to the head of the department and convener of this international e-workshop, Professor S. Krupanidhi from VFSTR, for his valuable guidance and encouragement in all aspects in organizing the workshop. I should not forget to thank the organizing secretary. So he is the man behind this workshop, that is Dr. Abraham Pillai, and also the co-conveners of this international e-workshop, Dr. Indira, Dr. Nazanin Bobby, Dr. John Babu, Venkata Narayana, Dr. Ranganath Reddy, and Ms. Nagalakshmi, treasurer, who worked for making this workshop a successful one. So without the strength of the participants, this workshop would not be successful. So I would like to extend my deep sense of gratitude to the participants who are working as researchers, pharmacists, academicians, and also industry experts. I am sure this workshop certainly enhances the academic excellence and also accelerates the research in the area of machine learning applications in bioinformatics. I also thank the help, support, and cooperation rendered by both teaching and non-teaching staff of the Department of Biotechnology, VIGNAN'S Foundation for Science, Technology and Research. I am thankful to our university administrators, namely our chairman, beloved chairman, Dr. Lauratai Garu, the vice chairman, Sri Krishna Devarayal Garu, Chancellor Sir, Vice Chancellor Sir, Registrar, for approving this valuable workshop. So last but certainly not the least, I would like to express my utter sincerity to the governance of Vignan for appreciating our efforts and providing us this opportunity. Thank you all. Thank you.
Oh, thank you.
Thank you, sir. Thank you so much, sir. Thank you to one and all for attending.
Yes. Feedback links will be shared via registered emails. To your registered emails, you just fill it and you can get the certificate within 24 hours. No need to worry. If anyone can, you should not get the certificate, you can reply to my mail, mladdba@gmail.com. No need to worry. Okay, you all will get the certificate.
Abraham?
Yeah, Kalyani.
Yes, please announce to all the audience that whatever lectures are there, they will still be on the YouTube. Plus, if you want to repeat it or you want the studying material, it will be shared on the GitHub links. So along with the certificate, we will constitute an email and we will send you all the details. So you're more than welcome to revisit it and practice it out.
Yes, ma'am. Yes. And in future also, we like to continue this kind of training sessions, ma'am. I already mentioned in the previous mail. I asked the participants to subscribe to our channel, Camerada, and we're going to deliver some sort of lectures regarding this molecular dynamic simulations, artificial intelligence, these sort of things.
Yes,
Yes,
yes, of course, of course.
We also instruct the
So
participants.
we are looking forward to the feedback from the participants, and whatever you need, we will constitute the next workshops according to that. We have some ideas, but we would love to hear from you, because ultimately we are doing this all for you. So your feedbacks and your advices are more than welcome, and we would love to hear from you, and it's very important. So thank you so much.
Thank you so much, ma'am. Yeah, Abhijit.
Sir, I want to inform you one thing.
Yeah.
I have received the mail regarding the feedback form. Sir, I think it is editable in our side. You may just cross check.
Okay.
Yes, Abraham sir, like even the same suggestion from my end, it is in editable format. You should have to share it in a link format so that you other cannot edit on one's information.
No one can edit other than you guys.
Yeah, yes.
Yeah. Other than yes, it's restricted. Other than you guys, no one could able to edit it. Okay.
But as of now, it is not in that posture. It seems
it's
like
in that position only. Among you only you can edit. Among you guys, 60 to 70 participants only you can edit. If you want the privacy, okay, I can do that. It's restricted only, na? Did you open it?
We have opened it, but it's something like other than the option that has been provided for filling up the name, email ID, and all, it is directly going to the edit format. Something like it is in a process of creating the Google form instead of filling it out.
Okay. No worries. I'll remove that and I will share it again. Okay.
Thank you, sir. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, Abraham. Thank you, participants.
Thank you, sir.
Thank you, sir.
Thank you, Abraham.
Thank you, Abraham, for organizing nice workshop.
Thank you, Abraham. See you next online workshop.